Cells of the immune system, T lymphocytes. Just like B lymphocytes, T lymphocytes are also uh, in complete surveillance in the whole body for foreign antigens or even for antigens that have been developed because of cells becoming cancerous. So these T lymphocytes are always on the alert for recognizing and then giving rise to an immune response that will help in eliminating the antigens. So let us look at the learning outcomes of the session. So the T cells are obviously lymphoid cells, just like the B cells are, and they originate in the bone marrow just like B cells, but the B cells mature in the bone marrow and the periphery, that is the secondary lymphoid organs, while the T cells are specifically maturing in the thymus gland. And the development and maturation of the pre-T cells gives rise to several subsets of T cells. The T cells circulate as quiescent cells or what you call as naive T cells, but they get activated when they interact with an antigen. But that antigen is MHC restricted, which means that the T cells can only recognize an antigen that is exhibited by major histocompatibility complexes. Now, these MHCs can either be expressed by antigen presenting cells, and those are generally MHC class 2, or any cell, any nucleated cell can also give rise to MHC class 1, and so antigens restricted by MHC class 1 can also be recognized by a subset of T cells. The T cells are able to generate an immune response that can eliminate antigens through humoral mediated immunity, that means by secretion of antibodies, cell-mediated immunity, which means that cytotoxic cells, and innate immunity, which means cells which are pro-inflammatory or cells which are phagocytic in nature. Of the many subsets of T cells, CD4 plus T helper cells and CD8 plus T cells are links between innate and adaptive immunity, with both, that is CD4 plus and CD8 plus, generating or having the capability to generate memory, immunologic memory. And that's the reason why it is part of the adaptive immune system. Now, when you look at the development of T lymphocytes, the T lymphocytes develop from hematopoietic stem cells. These get differentiated into what is called as common lymphoid progenitors, and the common lymphoid progenitors can then get, when they get into thymus, so this happens in the bone marrow. From bone marrow, the common lymphoid progenitor travels to or is transported to the thymus gland. Within the thymus gland, what is formed is the early thymoid progenitor, thymus progenitor, and this then gets differentiated into what is called as double negative pre-T cells. Now, what does double negative pre-T cells indicate? That is, when you get to look at the subsets of T cells, you will find that the major T cells are CD4 plus T cells and CD8 plus T cells. So the double negative pre-T cells neither have CD4 plus nor have CD8 plus at this stage. It gets further differentiated into double negative 3 type, and this double negative 3 type can give rise to a subset of T cells which is called as gamma delta T cell. Now, this gamma delta T cell has on its surface a T cell receptor which is made up of subunits gamma and delta, and that's the reason why they are called as gamma delta T cells. Now, these pre-T cell receptors can, these T cells can get further differentiated such that you have double positive pre-T cells being formed, and these double positive T cells would have both CD4 plus and the CD8 plus surface molecule. However, as it gets further differentiated, one observes that either CD4 plus from the T cells is lost or CD8 plus from the T cells is lost. So you may have a subset of T cells having only CD4 plus on its surface, or you may have a subset of T cells which only has CD8 plus as on its surface. Therefore, eventually what we understand by SP over here means single positive T cells. Therefore, effectively, you would have CD4 plus T cells that are being formed and CD8 plus T cells that are being formed that can go into the blood cells, and also the gamma delta T cells that can go into the blood and get circulated and go into the periphery into where you have the secondary lymphoid organs. So in the secondary lymphoid organs, when you have these released from thymus, they exist as quiescent or what is called as naive T cells. That is, these have not been yet exposed to an antigen. When these get exposed to an antigen in the periphery, in the secondary lymphoid organs, then you would have these T cells getting differentiated into effector T cells and memory T cells. So this is how the T lymphocytes get developed. One must also note over here that through the different stages, as in the T cell development, as the cells go from one stage to another, one can very clearly see at every stage what are the surface proteins that are present, and those surface proteins can actually be the signature for the T cells at that stage. So for example, when you have CD4 plus T cells, you know that that is one subset. When you have CD8 plus T cells, you know that that is another subset, because CD4 plus will not be present on the other, or CD8 plus will be present only on a specific subset. So this differentiation can lead to specific markers being present on the T cells, thereby demarcating one subset of T cell from the other subset of T cells. Now let us begin with CD4 plus T helper cells and understand a few aspects about CD4 plus T helper cells. So as the word suggests, these T cells have on its surface cluster of differentiation 4, that is proteins present on the surface. So it has CD4 and plus indicates that CD4 is present on the surface of the T helper cells. Second question that should arise is, why are we calling it helper cells? Why are we calling it T helper cells? So what has been observed with respect to the functionalities of CD4 plus T cells is that these help B cells to get activated and function. These help CD8 plus T cells, which is the second subset of the T cells, to get activated and function. It helps in what is called as class switching. So suppose a B cell has on its surface IgM, and due to the CD4 plus T helper cells, the IgM can get switched to IgG. So that becomes class switching. It can also help forming memory cells. So whether you consider helping CD8 plus T cell, or you consider class switching, or you consider forming memory cells, these all are associated with what we term as adaptive immunity. So only when the CD4 plus T helper cells are exposed to an antigen will it try to activate immune cells that are able to specifically fight out or eliminate out that antigen with which it has interacted. So therefore, adaptive immunity is always highly specific. One can also see that because it is activating CD8 plus T cells, and CD8 plus T cells are cytotoxic T lymphocyte, that is, they are able to bind to a target cell and destroy the target cell that contains a virus or that contains a bacteria. So they are phagocytic in nature, and therefore that falls under cell mediated immunity. Because they help B cells, and the B cells get activated and produce or secrete antibodies. Antibodies are part of humoral immunity. So you can see how CD4 plus T helper cell is contributing to humoral immunity. Also interestingly, CD4 plus T helper cells help in activating macrophages, neutrophils. They also produce gamma interferon, and we all know that gamma interferon is having an antiviral property. So in all of this, you can see that the CD4 plus T helper cell is helping in inflammation, and so these all contribute to what is called as innate immunity. So you must understand that innate immunity is able to be is basically a general immune response where it can act on any foreign antigen, whereas adaptive immunity is where it can act only on a very highly specific antigen. So therefore, you can see that how CD4 plus T helper cells are associated not just with innate immunity, they're associated with adaptive immunity, and with adaptive immunity also they're associated with both humoral as well as cell mediated immunity. Now we go to further understanding what are the structural aspects of CD4 plus T helper cells. So CD4 plus T helper cells, one major component of it is what is called as the alpha beta. So you can see this is the alpha subunit and this is the beta subunit of the T cell receptor. So this is the antigen binding receptor. But this antigen binding receptor, or what we call as T cell receptor, is always associated with a complex that is called as a CD3 complex. So it always exists as TCR CD3 complex, and this TCR CD3 complex can bind to an antigen, but that antigen has to be bound to an MHC. So it is always MHC restricted. Then other than this, which is a very key component of CD4 plus T helper cell, it has CD28 that can bind to B cells. You have CD2, you have LFA1, which comprises CD18, CD11B, CD45, CD54, CD4 you can see is the protein marker of the T helper cells. And this is able to bind to the target cell, allow binding to the target cell as well. So it's co-stimulatory as well. So these are certain very specific surface proteins that are associated with T helper cells, and each of these contribute to the various functions that the CD4 plus T helper cells carry out. Now let us see how when naive T cells interact with or are exposed to antigens, what or what does it differentiate into, or what does it get converted into. So a naive CD4 T cell can get differentiated into Th1 subset, so T helper 1 subset of the T helper cell, and this is a subset that is able to produce gamma interferon. We all know that gamma interferon is instrumental in restricting the spread of viral infections, now or even intracellular bacteria for that matter. So how does it act? The gamma interferon is released by the target cell or by the Th1 cell per se. It can induce the release of gamma interferon. Gamma interferon would actually signal all the other cells that are close by to prevent the viral replication. So if the viral DNA replication is prevented, the spread of virus is contained, or the spread of virus is restricted only to a target cell and cannot go on to the other cells surrounding it. So Th1 helper cell is the one that is going to help CD8 plus T cells, and so it is responsible for cell mediated immunity. Now, the Th2 subset of the naive CD4 is releasing other cytokines like IL-4, IL-5, IL-13. And these IL-4 and IL-5 are able to activate B cells and lead to what is called as humoral immunity, because the B cells on activation are going to produce antibodies specifically for the antigen that the naive CD4 has encountered. So that is again what is suggestive of the fact that one subset is specifically for humoral mediated immunity and one subset is specifically for cell mediated immunity. So this T helper cell is going to activate CD8 plus T cells, whereas this T helper cell is going to activate B cells. Now, naive CD4 plus cells can also get differentiated into Th17 subset, and this Th17 subset produces IL-17, and this IL-17 is associated with mucosal immunity as it is able to fight extracellular bacteria and fungal infections. One another subset of the T helper cell is what is called as T follicular helper cell, and this is able to control or contribute to the control of all types of pathogens, whether it is virus, whether it is bacteria, whether it is fungi. The other subset that is being activated is what is called, or getting differentiated into, is what is called as a T regulatory helper cells. And these T regulatory helper cells are associated with IL-10 and TGF beta, that is tumor growth factor beta. These are cytokines produced, and these seem to have a role in in suppressing immune responses. So therefore, T regulatory helper cells have been associated with preventing autoimmunity, while these are able to activate immune cells that contribute to innate immunity. And these, therefore, you can see how the CD4 helper T cells CD4 plus T helper cells are helping in various immune responses. Now, we go to the CD8 plus T cells. So, the CD8 plus T cells are MHC class 1 restricted. Yes, for that matter, CD4 plus T helper cells are restricted by MHC class 2 presented antigen, and CD8 plus T cells are restricted by MHC class 1, which means that the T cell receptor of the CD8 plus can recognize an antigen that is bound to MHC class 1 and not to MHC class 2. The responses are definitely through production of cytokines, so they are able to produce specific cytokines, and they are also responsible for cytolytic attack, means they are cytotoxic in nature. So they are able to actually engulf these cells and lyse the cells. So when you look at CD8 plus T cells, CD8 plus T cells can bind to the target cell. So it has the T cell receptor. This is the alpha beta T cell receptor, and this of course is the CD8 ligand that it has. So you can see that the alpha beta T cells are able to bind to MHC class 1 with the peptide. This interaction that happens between the CD8 plus T cells and the target cells is going to stimulate the CD8 plus T cell to release perforins. So when it releases perforins, then it is going to give rise to the lytic mechanism. So cytolytic attack is what we are talking about. These perforins are protein complexes that are able to produce spores or form pores on the surface of the target cell. And it is also releasing what is called as granzyme B. This is serine esterases. And these enzymes are able to induce apoptosis within the target cell. So eventually you can understand how the CD95, when it binds to CD95 that is present on the target cell, that's co-stimulation, and the perforin and the granzyme can lead finally to lysis of the target cell and thereby eliminating this target cell. Now, why is this called target cell? Because within this cell is present either a virus or is present an intracellular bacteria. What it can also lead to is what is called as a non-lytic mechanisms, and these non-lytic mechanisms are aided by the kind of cytokines that it produces. If it is gamma interferon, it becomes antiviral. If it is IL-17, it is basically triggering the innate immune system to get rid of any extracellular fungi or bacteria, etc. TNF alpha again leads to removal of bacteria, etc. So you can see that you can have non-lytic mechanisms or you can have lytic mechanism. Interestingly, CD8 plus T cells on getting exposed to the bacteria can or to the antigen can also form several subsets. So you have TC1. This TC1 is associated with cytotoxic activity. TC2 is associated with immune suppression. You can see that it produces IL-10, and therefore this also can act as something that can prevent autoimmunity. And you have TC17, which is associated with mucosal immunity, more that of an innate immune system, but it has been interestingly found to generate memory cells of TC17, and therefore there is a debate as to whether the cytotoxic T cells are also are not just associated with innate immune system but also adaptive immune system. So these are the different subsets that are present for CD8 plus T cells. Now you also have, we know that every subset of the T cells have what is called as a T cell receptor. So the T cell receptor can either be made of alpha and beta. So most T cell receptors present on CD4 plus T cells and CD8 plus T cells are of the make that has one polypeptide that is alpha type and the other polypeptide that is beta type. Of course, you can see over here that every T cell receptor has a binding site for the antigen, and the binding site of the antigen for every T cell receptor present on the respective CD4 plus T cell or the CD8 plus T cell can bind to a very specific antigen. That's because of rearrangement that is happening in the T cell receptor, which one can associate with also the B cell receptors present on B cells. That means every antibody that is present on the B cell is able to bind to only a very specific antigen. Now you can see how the CD3 plus is present. So when an antigen binds to the T cell receptor, automatically the CD3 gets excited or gets stimulated, and these in turn are able to transfer or to give the signal to the cell, T cell, and this T cell then gets activated. Now, when you have the unliganded, that means when you have the T cell receptor not bound to the antigen, then you do not have a stimulation basically. But when it is associated with MHC that has an antigen bound, you can see that the structure of the T cell receptor changes because of the binding to the MHC with the antigenic peptide, and because of that you have a stimulation of the CD3, and this stimulation of the CD3 leads to formation of or gives rise to signal transduction that activates the T cell. Now we also know that development of T cell leads to a very specific subset of T cells, which is called as gamma delta T cells. It is called as gamma delta T cells because the T cell receptor is not of the make of alpha beta, but it is of the make of gamma delta. But what is most interesting to understand is that the gamma delta T cells are not MHC restricted. They can bind to antigen directly. They need not, they are not specifically going to bind to an antigen only that is bound to an MHC molecule. So that is something which is very different from CD4 plus and CD8 plus T cells. CD4 plus and CD8 plus T cells can only bind to an antigen that is already bound to an MHC molecule, but gamma delta T cells can bind to an antigen on its own without the antigen having been bound to the MHC molecule. Now, this image is basically talking about how the gamma delta T cell has a role in graft versus host, rejection. So they're talking about the gamma delta T cells responsible for graft rejection, and those which are able to kind of be of help and help the body um, to actually accept the foreign graft that is there. So in transplantation, the gamma delta T cells have been found to have an important role. But what is essentially being mentioned over here is that you have several different receptors that are present on the surface of the gamma delta T cell, which enables the gamma delta T cell to also have multiple functions. As you can see, it has T cell receptor, which is the gamma delta type. It has natural killer type surface molecule, and it has CD16. It can give rise to cytokines, so it can also give rise to gamma interferon. It can give rise to IL-4, which can lead to regulation. You can have immune suppression. All of this is associated with gamma delta T cells as well. So it has natural cytotoxicity. This is important, and therefore gamma delta T cells have been used for therapy. It has been used or have been taken up and have been modified to be used for various purposes. It has immunosurveillance receptors. That means just like B cells, just like CD4 plus T cells, just like CD8 plus T cells, they can circulate in the body looking for antigens. They are able to produce cytokines, chemokines, and they are able to help B cells as well. They are therefore part of innate and adaptive immunity. They are pro-inflammatory, so that's the reason why they are associated with innate immunity. They are having the ability to present antigens, so they themselves can act as antigen presenting cells, and they are potential cells for therapy, especially cancer. So therefore, this has, although this is not very abundantly found, but it is found to be rare and aggressive as a T cell. So let us make the conclusions. The T cells arise from hematopoietic stem cells in the bone marrow, mature in the thymus with clonal selection to give rise to naive T cells. The naive T cells, on interacting with the antigens, bound to MHCs through the T cell receptor get activated. So we are saying that naive T cells on binding with the MHC restricted antigen get activated, they proliferate and differentiate into effector cells and memory cells. The CD4 plus T helper cells and CD8 plus T cells are molecules that can be part of both innate and adaptive immunity. It's not molecules, cells. Humoral as well as cell-mediated immunity. The same has been observed with gamma delta T cells. The CD3 TCR complex on binding to antigenic peptides get cross-linked, giving rise to a signal transduction pathway that can help in an effective immune response. So T lymphocytes are cells ready for a battle with intracellular as well as extracellular pathogens, with the responses being either general or highly specific. And these highly specific responses can be consolidated as a memory for future battles. Thank you.